afternoon or good morning. Depends the place of the world where you are located right now. But I am proud to belong to this society, ANR. I was one of the founders. I have the privilege and the honor to be invited for Dr. Wang to be one of the presenters. I am not only proud, but also I am very grateful to this society because during many years have been my spiritual support to continue doing this work. And that I believe is a relationship that we share with many of you. And say thank you, and thank you to Anne to invite me to give this talk. For me, it's a matter of grateful to say thank you, and also to remember our colleague that now is seeing us from a better place and was the big inspired of the whole Poland working in neurorestorology. Well, saying that, let me introduce our topic. It is a general presentation, the one that could serve for any type of tissue repair, because we consider that the influence of the immune system is essential to repair any kind of tissue, which is not an original idea of me. It not also was described for the ancient cultures like Assyrian, Egyptians, but also Hippocrates' first writings. Now, what is going on here is inflammation is necessary to promote repair of any tissue. Now, how that could be concentrated in modern language? We can summarize in the following way. After an injury damage of any organ, there are a common pattern of sequence of events to promote tissue's repair. But if this process is insufficient, it led to the scar formation or its fibrosis. Injury occurs, damaged tissue produces chemotoxic substance to mainly attract PMN to the area. Polymorphonuclears invade the area, start to clean cells and tissue debris, as well as adventitious microbes, attracting monocytes to the area. Monocytes continuing cleaning the area and producing chemotoxic signals that attract local and distant stem cells, as well as elicit an organ-specific lymphocyte activation. Effector cells and stem cells start the stroma and parenchymal repair. But if the process is exceeded for the properties of the wound, the same process led to the development of a tissue fibrosis and or scar formation. Now, that is a schema with a little bit more sophisticated way are saying the same that I said in the last diapositive. But mainly we can say here that we have the first 2 days with acute inflammation oxidative phase, and that is no good to do any kind of tissue repair, cell repair strategy. There is a second period that is between the day 3 and day 12 with subacute inflammation that produces a pro-regenerative phase and stem cell proliferation and differentiation. Therefore, afterwards, there is a third period dominated for chronic inflammation, and that is a profibrotic phase. The cell differentiation is complete, not started, but also the stroma cells start to produce myofibroblasts that are the responsible for the scar formation. And that happened also in the site of the spinal cord wound site. Now, that is a general schema where I added this last line, and I said, beside effector cells have all this immune already recognized effector function. There is an effector function involved in the stem cell differentiation and also T regulatory cell promotion that is also an effect of the effector cell. Now, let me explain a little bit more this last cell line. What happened with this new kind of effector cells? Now, let me say, if I take a bunch of mononuclear cells from the bloodstream And I put those in presence of an antigen, in this case, brain antigen, like B cerebrolysin, that produce effector cells because they induce for the already existing effector cells against the central nervous system an amplification of cells that are directed against central nervous system. However, there is a parallel phenomenon that occurs, and these effector cells also produce different epigenetic factors, Herbigracia neurotropins, that induce stem cells to differentiate into neurogenic neuroblast-like cells. Effector cells are attracted to be in touch with those stem cells. It is a chemoattraction and receptor-mediated intracellular phenomenon that develops gap junctions in between both cells and different interchange of paracrine factors. As a result of this physical and paracrine crosstalk, stem cells became neural progenitor cells, and effector cells became deregulatory cells. Now, how we did it? We put it in a bunch of mononuclear cells, the antigen, in this case cerebrolysin, and only the cells that previously have been activated against central nervous system, and we have that in all our normal bloodstream, 1 in 10,000, they amplify, and the other one pass away, they die. Therefore, we get a concentration of those cells. If we put those cells into the tail of a nude mice, we can observe like this positive tonal reaction. That means those cells are specifically attracted to the brain, and there they produce neural death. Now, if 2 days after we have done this injection, we injected Wharton gel in the mesenchymal stem cells, we can observe how those cells were stopped in their action and also promote with the Wharton jelly the already damage produced for the first injection of effector cells. What happened with the cells itself? We try to analyze if they produce any kind of paracrine function. We also observed that previous to the in vitro incubation, mononuclear cells didn't produce any kind of neurotropin. At 72 hours, when they are activated, they became positive for at least these 3 different kinds of neurotropins. Therefore, we are seeing here changes in the nature or the expression of genes in the lymphocytes, but also in the mesenchymal stem cells that became neural progenitor cells. Why we say that? Because they start to appear what we call it specific protein synthesis. A gene that activates start to produce a specific RNA, and they produce a specific protein. And that could be seen here because after the co-incubation, those mesenchymal stem cells not only change morphologically, as we can see here, but they also became positive for nestin, triiodopyrin acid protein, NEO66, and tubulin 3. If you remember from our biology studies, that is a chromosome from the Drosophila, and you can detect here the poof of those chromosomes, where the active sites where the Drosophila chromosome produce RNA messenger. And you can see in the same chromosome at different stage of development of the larva into fly, you can see how the same gene is activated, blocked, activated, blah, blah, blah. The same locus happen the same. That is something that we need to observe in our model to be knowing that that is a sequential phenomenon that happens in effector cells. And that happens really. If we co-incubate cells against muscle in presence of mesenchymal stem cells, the first 21 hours, the only genes that they show it are the one named MIF5, that is the first precursor gene. The next day, you hardly will find any positive MIF5 positive cells, but you are going to be able to identify that those same cells are producing now MyoD in a larger quantity. And without ending to produce MyoD, they, the third day, start to produce myogenin. Is this up and down regulation of those genes that show us cell differentiation happened in the short time of 3 days, when the simple change in vitro to make this differentiation for muscle cells take at least 10 days, and for neural system take more than 2 to 4 weeks. Same happened with cells that try to become cartilage progenitor cells, okay? But, and those are the different assays. You see how SOX9 and GDF5 that are essential at the beginning. SOX9 is present in MSC and continue to be present in the following days of differentiation, but GDF5, that is the first gene that really directs the cartilage differentiation, appear only 24 hours later and then is downregulated. And at the same time, other proteins that are important, like the COMP and pregabalin 4 and collagen type 4, all start to grow up. That means a sequence. We can say the first day only we see SOX9, previous to that, GDF5, that is the first indication of cartilage differentiation, and finally start to appear all these substances that are the ones that really tell you we are already obtaining the differentiation. This sequence process we can observe it here. Now, to emphasize the necessity of those effector cells in vitro, we have developed the following experiment. We have injected those some wild rats with a caustic substance named monoiodoacetate. When we injected those substances with monoiodoacetate, you may observe that 7 days after, they have a terrible destruction of the cartilage. And if that day you give it to them, because it's the time where the maximum point of inflammatory cells happen, you give them different type of biological treatment, and you kill the animals 3 weeks later, that means 4 days after the destruction of the tissue, we can observe different degrees of repair. Let me show you here. That is the control. You see this small thing is the only places where you can observe some cartilage. And that keeps at 7 days, 28 days, and 50 days. Very poor spontaneous regeneration or non-spontaneous regeneration. Now look at how different from this image of control at day 28. You can see marked for the yellow line the size of cartilage. Okay, you see the only addition of MSC produces some increasing. There is also some increasing with effector cells alone, but see how huge different is the synergistic effect. It's farther than the summatory. It's a potentiation of both. The action of both together, effector cells and mesenchymal stem cells, really produce earlier. Now, see what happens when we alter the inflammatory condition of the host. Now, that again is the control. That again is what happened with the combination of both using rat cells or rats. Now, see what happens if we use human cells in rats. There are huge, those huge that I am pointing here and is signaled by IT. Okay, those are huge inflammatory panels reaction, connective tissue, inflammatory connective tissue, with very poor response of regeneration. And if we use new rats with the same human cells, even when we give it all the effector cells, we have a minor response of regeneration, if not null. We have not inflammation, but also we have not possibility of repair, because those animals lack the host response for the inflammation. Finally, when we have been using this schema of therapy in humans, and that have been done for the last 6 years, actually we developed this protocol in conjunction with a colleague of Dr. El Masri, our good friend that was the head of the Department of Orthopedic, Dr. Richardson, and unfortunately 2 years ago he left us because he passed away for a sudden myocardial attack. Now you can see here, okay, in this patient, that was one of our first patient treated after we did the corresponding animal experiment. He has no subchondral bone, and how he has recovered the subchondral bone. He has not cartilage, even the hole here, and how doing a biopsy in the same place, we see a complete regenerated cartilage and a complete subchondral bone. We believe that this interaction between effector cells and mesenchymal stem cells, or any kind of stem cells, even we have proven that right now, when I now presented those results, that also placental or white progeny origin effector cells could have the same phenomena for different type of organs. And that is a general condition that should be taken into account when we plan further cellular therapy strategies to solve spinal cord injury, brain injury, or any kind of organ injury that could be similar to this condition.
I wanted to say thank you very much for the attention and the timing again that you have given to me to express this, what we believe are the fundamentals of all our theory. Now you are going to see Dr. Moviglia showing you our results that are the follow-up of many of the patients that we have been showing in the past presentations. But what is interesting about those patients is that they have been progressing without any cellular therapy after 2.5 years. And that really talk about not only the efficacy, but also the efficiency of the treatment. Not only is it good to promote the acute repair of the tissue, but this repair could be sustained for a longer time. And that is what really we wanted to share with you, because it's our proud, but also I believe it's also a common achievement of the whole group. Thank you very much.